Good morning, dear students of grade 10. Welcome back to our session. In photosynthesis, so far we have discussed about what is photosynthesis, why it is called a photochemical reaction, then the factors essential for photosynthesis, external factor, internal factor. The external factors such as carbon dioxide and sunlight, whereas the internal factor chlorophyll and of course water. These internal, external and overall, all the four factors essential for photosynthesis. For a green plant to perform the photosynthetic activity, these four essential factors we learn. Even we learn chloroplast, a type of plastine which contain green color pigment. The different pigments, chlorophyll A, chlorophyll B, carotenoid, xanthophyll, all other accessory pigments which plays a major role during photosynthetic activity. All these we discussed. Then further we discussed about structure of the chloroplast, grana, stoma, thalappoid. Then the important phase of the photosynthesis we enter, that is light reaction and dark reaction. The light dependent reaction, what we learned, the formation of ATP and NADP during the light dependent reaction, and the end products of light reaction ATP and NADP, which are further used for the fixation of carbon, and carbon dioxide fixes further and it converts into carbohydrates, which is our glucose, that we learn during light independent reaction. After learning all these, we are into the next phase of photosynthesis, that we are all aware that the chemical equation which represents the photosynthesis process, the photosynthetic activity in green plant. Now, see, this is a chemical equation: 6 carbon dioxide molecules plus 12 water molecules, 6 CO2 plus 12 H2O gives rise to C6H12O6. This is the major product. Then in addition to this, 6O2 plus 6H2. This chemical equation represents photosynthetic activity in green plant. With the help of this chemical equation not only shows us the formation of glucose, carbohydrates from this, but also it indicates the minimum requirement for photosynthesis. As we know, 6 molecule of carbon dioxide which enters through the stomata in green plant, and 12 molecules of water. Of course, the amount of quantity of water is existed in a plant body before itself, as the cytoplasm of every cell has water contained. So these 6 carbon dioxide molecules, they react with 12 molecules of water. Both where they meet, they meet in a green pigment, chlorophyll. They meet in a chlorophyll in presence of sunlight. The entire process occurs in presence of sunlight, is it? So this is what our reactant 1. The reactant 1, what we have, 6 molecules of carbon dioxide, and the reactant 2 is 12 molecules of water. 6 carbon dioxide plus 12 water. When they both react in a chlorophyll, they both meet in presence of sunlight. Who will trap the sunlight? Chlorophyll molecule trap the sunlight. And due to the reaction between these 1, 2 and 3 in presence of sunlight, there is a formation of C6H12O6. This is our carbohydrate or glucose. This is our product 1. How many products are formed? The major product form is this one. And in addition to this we can say byproduct, product 2, and another byproduct water, that is water. Product 1 plus product 2 plus product 3. So altogether we are getting 3 products during this reaction from 2 reactants. Reactant 1 plus reactant 2 gives us these 3 products. So this chemical equation denotes, indicates the formation of carbohydrate from carbon dioxide and water. In addition to the carbohydrate, glucose, what is showing? 6 oxygen also liberated, given out. You can put this aroma, upward aroma in front of this oxygen molecule. That indicates, that denotes oxygen is evolved, oxygen is liberated during photosynthesis process in green plant. So here our experiment, the title of this activity is the experiment to demonstrate the oxygen production during photosynthesis. This happens in a green plant in presence of sunlight in a chlorophyll. This we learned. Now, what is our main objective, main theme is to demonstrate the oxygen production during photosynthetic activity. As shown in the figure, to demonstrate this, what are the requirements we need? We need test tube, then we need a beaker. The number 1 requirement is test tube, then beaker, then the number 3 is funnel, tissue funnel is placed in a beaker. Then we need hydrilla twigs. These are the requirements 1, 2, 3, 4 as shown in the diagram. To prove this, we say that during this photosynthetic activity, when carbohydrates are formed from carbon dioxide and water in presence of sunlight and chlorophyll, oxygen are given out. This shows that oxygen is liberated, oxygen is evolved. That oxygen is given out, that can be proved, demonstrated by this simple experiment. For this experiment, what are the requirements we have? We have to take a test tube, beaker, funnel, and hydrilla twig. Why the hydrilla twigs are preferred here? Because since hydrilla is an aquatic plant, it grows in water, it's a hydrophyte. The rate of photosynthetic activity in hydrophytic plant, hydrilla aquatic plant, is much faster, greater than other terrestrial plant. Therefore, hydrilla is preferred here to demonstrate this. Look at this here. See here. This is the test tube we have. Test tube. We have taken the beaker. The beaker is filled with water. See here. This is a beaker. The beaker we have a 20 ml beaker. We fill it with water. Then we have taken the funnel. In a funnel, a small tissue funnel is taken. In a funnel, what we have inserted? Twigs of hydrilla. Then the water containing beaker, we have to insert the hydrilla twigs in the funnel. Then the funnel to be placed in the beaker containing water in this way. Slowly we have to keep it, place it down. Then take the test tube. We have to invert the test tube over the funnel. We have to invert the test tube over the funnel like this, in this way, as shown in the diagram also. And after setting up the apparatus in this way, this entire setup we have to place it under sunlight. When the entire setup is placed under sunlight, what happens? We start, when we set up the entire apparatus in this way, beaker is taken, water is filled in the beaker, then hydrilla twigs are inserted in the funnel, then the funnel is placed in the beaker containing water, then the test tube is inverted over the funnel, then the entire setup is placed under sunlight. The moment after a few minutes, we start observing the air bubbles coming from the hydrilla twigs, and they start moving towards the aerial part, towards the top of the test tube. The droplets, the air bubbles, they move and finally they go and collected at the top there. And that is what we say, the bubbles which go and finally they get collect here at the end of the test tube, that we say oxygen. How can we say that is an oxygen? After this, we have to remove the test tube slowly. If the burning splinter is introduced in the funnel, test tube, the moment you introduce a burning splinter in the test tube containing oxygen, the burning splinter burns brightly. It burns brightly. Since oxygen is a supporter of combustion, that burns brightly. If it do not burns brightly, the burning splinter, we have to conclude that there is no oxygen. And here the oxygen is evolved, the oxygen is present in the test tube. The moment you introduce a burning splinter in the test tube, it burns brightly. That indicates oxygen is present in the test tube. And from where the oxygen has come? The oxygen has come from during photosynthesis. So this type of simple experiment help us to demonstrate that oxygen production during photosynthesis process. So the next process, the next activity will be taught in our next class. So we'll stop here. Thank you.